Hello everyone, welcome back to another video. This is AP Biology Unit 1, Chemistry of Life, reviewed in 5 minutes. 2027 Comprehensive Review for Scholars for Success. 1.1 is Structure of Water. It is understanding the polarity and hydrogen bonding as the engine for biological organization. Oxygen is partially negative, while hydrogen is partially positive. For this reason, the partially negative will form a covalent bond with the two partially positive hydrogen atoms. Oxygen's high electronegativity creates an unequal distribution of charge, which creates this polar covalent bond. Intermolecular forces forms between these water molecules in a form called hydrogen bonds, which forms between the partial negative oxygen of one molecule and the partial positive hydrogen of the other. The emergent properties of water include cohesion and adhesion. Cohesion is between two water molecules, and adhesion is between one water molecule or multiple water molecules and another polar substance. These two combined allow for water to cling to cell walls of plants, also called capillary action. The second is thermal regulation. High specific heat allows water to resist temperature changes, absorbing significant energy before increasing molecular movement. The third is solvent versatility. The polarity of water allows it to dissolve ionic and polar substances, creating the aqueous environment for metabolic reactions. Additionally, water cannot dissolve non-polar substances. The final emergent property of water is density. Ice is less dense than liquid water due to the crystalline lattice of hydrogen bonds, insulating aquatic life during freezing conditions. This is why ice floats on top of any water body. 1.2 covers the elements of life. The primary elements that make up all organic molecules include carbon, hydrogen, nitrogen, phosphorus, and sulfur. Carbon's role is the backbone of all organic molecules, and it is present in carbohydrates, lipids, proteins, and nucleic acids. Nitrogen is essential for building amine groups, and it is present in proteins and nucleic acids. Phosphorus is critical for energy transfer, and it is present in nucleic acids and phospholipids, which are a type of lipid. Sulfur forms disulfide bridges for folding, and it is only present in proteins. Note: CHNOPS represents the 6 key elements required for a complex biological organization. Memorizing that acronym is helpful for exams. Biological macromolecules, 1.3 to 1.7. Structure, function, and information storage. The classes of macromolecules include carbohydrates, lipids, nucleic acids, and proteins. The monomer of a carbohydrate is a monosaccharide, and the use of a carbohydrate is quick energy and structural support. Lipids do not have a direct monomer, but their closest monomer is nonpolar hydrophobic molecules, and their primary job is long-term energy and membrane structure. Nucleic acids monomer are nucleotides, and their job is to encode, transmit, and express genetic information. Proteins monomer is amino acids, and their primary job is to execute cellular functions and catalyze reactions. Protein conformation includes the hierarchy of protein folding: primary structure, secondary structure, tertiary structure, and quaternary structure. Primary structure includes the linear sequence of amino acids. Secondary structure includes alpha helices and beta-plated sheets, connected through hydrogen bonds. Tertiary structure includes R-group interactions, including ionic, hydrophobic, and disulfide bridges. Quaternary structure includes the aggregation of multiple polypeptides. Function is strictly determined by structural conformation. Atomic proportions in humans include oxygen, which is 65%, carbon, 18.5%, hydrogen, 10%, and nitrogen and others at 6.5%. Despite being carbon-based, oxygen is the most abundant element by mass due to its presence in water, which makes up a significant portion of the human body. pH and enzyme stability are directly related. Enzyme activity is optimal at a specific pH range, and two extremes, both low and high, cause the enzyme to denature, which would result in the protein changing its shape and losing its biological function. Thank you for watching today. If you would like to see a more in-depth review, check out my channel where I cover each individual chapter of Unit 1 and on in much more depth.